Capillary electrophoresis is a laboratory separation method. Separate ions and molecules toward opposite poles through narrow capillary tube under electric field, based on electrophoretic mobility, as size, and charge, and hydrophobicity, as mentioned in next slides. Continuously. Capillary electrophoresis. Separate proteins, peptides, amino acids, nucleic acids, inorganic ions, organic ions, and whole cells, as mentioned in next slides. Capillary Zone Electrophoresis. Migrate ionized molecules into distinct zones to analyze amino acids, peptides, proteins, and other ionizable molecules, as mentioned in next slide. Capillary Gel Electrophoresis. Separate nucleic acids as DNA and RNA molecules, and proteins, as mentioned in next slides. Capillary Isoelectric Focusing. Separate amphoteric molecules as peptides and proteins, based on isoelectric point, as mentioned in next slide. Capillary Isotachophoresis. Separate DNA fragments and other biological macromolecules, based on electrophoretic mobility, as mentioned in next slide. Micellar Electrokinetic Chromatography. Separate all ions. Use surfactant in running buffer above critical micelle concentration, as mentioned in next slide. Continuously. Capillary electrophoresis. Have power supply to generate electrical field for separation and buffer reservoirs, which are two identical tubes filled with anodic and cathodic solution to ensure stability of electric field. And electrodes, which are cathode and anode, linked to power supply and buffer reservoirs to generate electrical field across capillary and capillary, which are fused silica fewer than 100 microns, act as separation channel and optical viewing windows to observe separation process. And injection system, used to load samples and buffers by gravity, or pressure, or vacuum, or electrokinetic, and detector, used to monitor amount of substance passing through capillary. And thermostatic system, maintain constant temperature inside capillary to ensure stability and accuracy. And recorder, used to record data after complete electrophoresis operation, and computer, collect data for analysis, as mentioned in next slides. Capillary electrophoresis. Initiate by dissolve samples in suitable buffer solution, and introduce samples into capillary tube, and apply electric field along length of capillary tube, for separation. And detect separation by detector, and record data in frame of electrogram, as mentioned in next slides. Therefore, capillary electrophoresis consider high efficient and separative power, because it requires small amount of samples to separate significant biological macromolecules, as proteins, peptides, DNA, RNA, and biopolymers. On the other hand, capillary electrophoresis consider labor and costly and time-consuming method, and sensitive in contaminants in samples and buffers. Finally, capillary electrophoresis use in genetic studies, and biochemistry studies, and disease diagnosis studies, and pharmaceutical studies, and food studies, and environmental studies, and forensics studies.